Hello everyone, welcome back to AS and A-Level Biology with Dr. Demi. I am Dr. Demi, and in today's video I am going to cover genetics. Now genetics is really broad, so this is not the only video that we will have, but I hope that this video helps you understand as we start to go along. This video has a lot of worked examples, so you're going to hear me clicking my mouse a lot because I'm trying to work through the examples with you and just explain how things work in genetics. But basically what this video will teach you is how genes are inherited, whether the genes are recessive or dominant, and all of those other things that you need to know. So if you have just found this channel, please note that this is the chronological order of the AS and A-Level Biology syllabus. We are now on chapter 16, and the hope is to finish the syllabus very soon and then to have recordings of past question papers so that you can see how to solve question papers and sort of improve your technique in answering questions. All right, let's get into genetics. So before we go into genetics, obviously we have to have key definitions, and I always put these at the forefront when I'm teaching my students in the classroom because I want them to just have them at the back of their minds as they go along. So when we speak about a gene, so for example, someone says, oh, I have good genes, or I love people with good genes. A gene is a length of DNA that codes for the production of a polypeptide. And when we say a polypeptide molecule, it's very important to note that that means a protein. We human beings are basically made up of proteins. Proteins are the ones that determine what enzymes we produce. Our enzymes are proteins rather. Our genes also decide how we look, and all of these things are all the results of proteins that are produced from the genes. An allele is something you will start to hear a lot as we go through this section of biology. And what an allele means is a different form of the same gene. So with alleles, you would have what we call a dominant allele, for example. A dominant allele is basically an allele that is always expressed, irrespective of what other allele is present. So let's say, for example, I'm just going to scribble a bit here in red. Let's say, for example, you have your dad has, let's say, black hair, and your mom has blonde hair. Okay. If an allele is dominant, we write it in capital letters. Okay. And if it's recessive, we write it in small letters. If these two exist in you as an individual, so say you have black hair and your mom has blonde hair, and these are the two alleles that they've given you for hair color, this combination will mean that you have black hair. You're not going to express blonde hair because black hair is dominant to blonde hair. All right, so black hair would be the one that that features. But say in future you decide to meet someone and that person also has the exact same combination as you. Okay, there is now a chance for the two recessive alleles to find each other, and that means that you might have an offspring with blonde hair. So this is just to tell you about how alleles influence what expressions of phenotypes we see across board, but don't stress too much about it. All of these become clearer as we go along. But always know that an allele is a different form of the same gene, and they can occupy the same locus on a chromosome or different loci on a chromosome. Genotype is what is the genetic makeup of an organism, and a genotype is often determined by alleles. So again, just using the hair example that I just did, where I said big B is for black hair and small b is for blonde hair. In this case, this is the genotype of this person. They have two different alleles representing the same trait. So the genotype is the genetic makeup and is determined by the alleles. Homozygous means that the two alleles for a trait are the same. So if I say, for example, that a person is homozygous for black hair, and we have all agreed that capital B represents black hair, then that person's genotype would be big B, big B. So that is what homozygous means. Heterozygous means that the two alleles for the same gene are different, and this would be heterozygous. Black hair, blonde hair, but the person expresses black hair because black is dominant. The phenotype is the observable expression, which means it's the physical appearance of a gene. So, for example, you might have a gene that has black hair and blonde hair. You might have those two alleles, but you're expressing black hair because that is the dominant allele, and so your phenotype would be black hair. I hope that is sort of clear. If you need any clarification, just post it in the comments. Just ask politely for an explanation, and I promise to respond to you. So yeah. So now, how do we inherit genes? So remember that when we started with meiosis, we were speaking about how at the end of meiosis, we have 4 haploid gametes, and each gamete will have 1 pair of chromosomes. So basically, each gamete has 23 chromosomes, okay, not 1 pair. It has 23 chromosomes for human beings. And so what you would find in that case is that with those chromosomes, each gene has alleles, and the alleles, like I said, can be dominant or they can be recessive. In some cases, you have alleles that are co-dominant. What this means is that both of these alleles are expressed at the same time. So again, dominant alleles will always be expressed even in a heterozygous organism. So even if an organism has 2 different alleles, the dominant allele would be the one that's phenotypically expressed. And if an organism has 2 recessive alleles, then the recessive alleles would be expressed if there are no dominant alleles. So bear that in mind. Dominant alleles are always expressed in homozygous and heterozygous organisms, but with recessive alleles, they are only expressed when there is no dominant allele present. Don't stress too much if all of these seem confusing. What I have done with this video is I've put together examples from the textbook, not examples, problems. And what we're going to do together in this video is just walk through those problems so that you develop a better understanding. Okay, and this is our very first problem. And you can see I've left lots of space so I can scribble with my pen over here. I'm just going to use a black pen this time around. So let's look at this. It says in mice, the gene for eye color has 2 alleles. The allele for black eyes is dominant, whereas the allele for red eyes is recessive. Choose suitable symbols for these alleles and then draw a genetic diagram to show the probable results of a cross between a heterozygous black-eyed mouse and a red-eyed mouse. Now, first things first, whenever you are asked in genetics to choose symbols, I always tell students to choose symbols that are not ambiguous, which means choose symbols that can easily be differentiated. So I always tell students, don't pick symbols like S, for example, because between capital S and small s, it might be difficult for an examiner to tell the difference depending on how you write. So always pick letters that are very easily differentiable between the one that's a capital letter and the one that's a small letter. So for example, you can pick the letter R, okay, because big R and small r look very different from each other. So that's just the tip that I'm sharing with you here. Let's get back to our problem. So here's the gene for eye color, and it has 2 alleles. The allele for black eye is dominant, whereas the one for red eyes is recessive. So let's say we decide we've been asked to choose the symbols, so we can decide to go with R. Okay, so that means capital R will be for black eyes because that's the dominant one according to the question, and then small r will be for red eyes because that is the recessive one. Okay. And then it says, draw a genetic diagram between the probable results of a cross between a heterozygous black-eyed mouse and a red-eyed mouse. So when they say it's heterozygous, again, remember what I said when we discussed key definitions. It means that it has 2 different alleles, but it is expressing the dominant phenotype. So heterozygous black-eyed mouse means that the genotype of this mouse is big R and small r. Okay. And a red-eyed mouse. Now, because red eyes is recessive, the only way a mouse would be red-eyed is if there is no dominant allele at all, and so that means the genotype there would be small r and small r. So now let me show you what a cross would look like in this case. So here you take this allele over there, that's big R and small r. Just going to draw a table here, and then you have small r and small r. These are the alleles that are combining. Now, something to say about crosses that you do in genetics. They are basically predictive measures. They're basically trying to show you what the chances are of obtaining certain phenotypes. So let's say, for example, here we go. We're going to cross this R and this R, all right? So here we have capital R and small r. This R times this R would be small r, and we repeat the exact same thing again, and then we have capital R, small r, and then we have small r and small r. What this is telling us is that we have, I just need to write this R properly, the way it looks is really bothering me. What this tells us here is that we have a 50% chance of having a black-eyed offspring, okay, and a 50% chance of having red-eyed offspring. This is what the cross looks like. And this is just an example of how you would do a cross, for example, in the test or in an exam. But don't worry, I have more examples for you. So if you felt confused by that one, we will do more. Okay, let's look at this one. This is a very popular question in my classroom. We take it from the textbook. Again, I just want to state that all the questions that we are using from the textbook that I have indicated I've been using in my very first video. So please just take note of that. And so here it says a species of poppy may have plain petals or petals with a large black spot near the base. If two plants with spotted petals are crossed, the offspring always have spotted petals. A cross between unspotted and spotted plants sometimes produce offspring that all have unspotted petals and sometimes produces half spotted and half unspotted. Explain these results. Now, my students usually get a chance for about 5 minutes to just walk through this together in groups, but let us try to do it together here in this video. So what this question is asking you to do is basically figure out which one of these plants have a dominant allele, or which feature is dominant and which one is recessive. So students just get about 5 minutes, like I said, to work on this, but it is really easy to figure out. So let's think about it. Let's read the information we've been given. And this is what I tell every student in genetics: read what you've been given and underline what's important. So we know that species of poppy may either be plain or they may be spotted. All right, so we can even choose our symbols now, and we can say, okay, let us make big B our plain, okay, and let us make small b our spotted. All right, I'm obviously not doing too well with annotations here, given how I'm leaving random spaces and also drawing random lines, but I hope that doesn't bother you too much and you just focus on what we need to get through. Okay, good. So big B is plain, small b is spotted. We've chosen symbols because we just want to see what happens. And then it says if two plants with spotted petals are crossed, the offspring always have spotted petals. So that basically means if we cross plants that are big B, small b times small b, all the offspring would basically be small b, so they would all be spotted because we have agreed that this allele is for spotted, or that's what we've chosen. The interesting part here comes up when they say a cross between unspotted and spotted plants sometimes produces offspring that all have unspotted petals. So all of them have unspotted petals, even when they are crossed with spotted plants. That suggests to you very quickly that big B, that means unspotted, might be the dominant allele. And sometimes it produces half spotted and half unspotted offspring, or half spotted and half plain. So now let's think about this. Well, if you want to answer this question with empirical evidence, that's what I'll call it, the best thing is to say, well, what would the crosses would look like if we were to do this? I'm just going to erase that because it looks weird. So we can say big B times small b, because this is now unspotted times spotted. All the offspring will be big B, small b. Now, because big B is for plain and plain is dominant, according to what we are choosing, all the offspring would basically come out unspotted, which is what this is telling us has happened. And then they say sometimes they produce half spotted and half unspotted. That might mean that if we take a heterozygous unspotted plant and we cross it with a spotted one, what we will have in this case, and I'm just going to draw it out as a table so that you can follow it and just see what I'm doing. So let's do that there. We're going to have, these would be unspotted over here, okay, and these here would be spotted. And so what this means is that our choices that we made at the beginning here are correct, that half unspotted, half spotted. You can see there, half of these are spotted and the other half are unspotted. And so we can then say confidently that the allele for unspotted is the dominant allele. And so that is how it plays out, and that is why it allows this to play out. Of course, you can try this out. I'm not going to do it with the reverse and just say, well, what if spotted was considered the dominant allele? And what you would find is that you would not be able to match the results that have been provided in the question. So that is just a good practice if you want to do that and check it out. Okay, now that we've whet our appetite sort of with crosses and, probably getting you all excited, just want to point out to you what we call generations and test crosses. So whenever you find questions in CIE exams, you might sometimes encounter what is called the F1 generation. And what the F1 generation simply means is that you have a cross between a homozygous dominant genotype and a homozygous recessive genotype. So if we were to use the example we've just concluded of spotted versus unspotted, in that case, if we were to say this is unspotted and it's homozygous dominant, remember that homozygous means that the two alleles are the same, and we cross that with the homozygous recessive, which in this case would be the spotted allele. We're going to end up with all of the organisms being big B, small b, and all those organisms would be considered the F1 generation. Now, the F2 generation is the generation that results when you cross two heterozygous F1 organisms. And so that just lets you know that this is what we call heterozygous. Heterozygous because the two alleles are different from each other. And so when we cross two heterozygous F1 organisms, we will end up with what we call the F2 generation. And just want to point out to you that from the F1 generation, you always have a heterozygous offspring because of the nature of the cross. So it's a cross that is a result of you combining a homozygous dominant, which is this one, with a homozygous recessive. So inevitably, you would end up with a heterozygous organism. All right. Test crosses are also something that's very important, and I've seen questions where students are asked to define a test cross. And a test cross is a cross between an organism showing a dominant phenotype and another organism that is homozygous recessive. And when we say a dominant phenotype, it simply means that with test crosses, we don't know what the genotype of a certain organism is. And so we use a test cross to determine that by crossing that organism with a homozygous recessive. And you use it to determine the genotype from that. And I think we have examples on that on this video. So just stay tuned and sit tight in your chair and let's see what we've got. Okay, so let's look at an example of a test cross. So this again is from the textbook. It says in Dalmatian dogs, the color of the spots is determined by a gene that has two alleles. The allele for black spots is dominant and the allele for brown spots is recessive. So we can just go ahead and choose our symbols now. And I know I'm using B a lot, but it just seems like it's just the easier one. So it says black spots are dominant and brown is recessive. Okay. And so that's B is equal to brown. So remember that with dominant, we usually use a capital letter, and for recessive, we use a small letter. So here, a breeder wanted to know the genotype of a black spotted female dog. She crossed her with a brown spotted male dog, and a litter of 3 puppies was produced, all of which were black. The breeder concluded that the female was homozygous for the allele for black spots. Was she right? So now let's look at how this would play out. Usually students try to do this in their heads. When I give it to them in the classroom, they try to, move the information around in their heads and say, okay, well, this is how I think about it, and that's great. But I always say the best way to get the good information is to put your pen to paper. So I hope that you're doing so as well as you're watching, but I'm also going to write it here on the slide. So let's look at this here. It says we have a black dog, black is dominant rather, and brown is recessive. And so we don't know the genotype of this black dog, this female black dog, but we know that the male dog is homozygous recessive and he has brown spots. So we know that the genotype of what was crossed here is small b and small b, and over here is just X. If that made you very nervous, this is not mathematics, although we do have some math in this chapter. This is not it, but just trying to let you know that this is the unknown over here. Cool. So the breeders concluded that the female was homozygous. Now let's have a look at that. If the female was homozygous, she would be homozygous dominant, right? So that would be big B. I'm just going to erase over here so that I can make it into a table. That would be big B, big B. And over here, we would have small b and small b, okay? And so when we do this cross here, all the offspring will be big B, small b. And because big B is black and it's dominant, that means all the offspring will be black. And in that case, we can then say, okay, we sort of agree. But let's think about the fact that what if the black dog was heterozygous, what would our results be? So that would then be big B, small b for her genotype. That's for this unknown dog, because again, we're just testing things out to see what could possibly be correct. All right, and then we cross her with a homozygous recessive male brown spotted dog, and we would have half of the offspring being big B, small b, and the other half would be small b, small b. And what this then tells us is that, okay, well, we don't have any brown dogs here because this represents brown. And so we can sort of say, okay, the breeder was correct. But also, if you want to think deeply about this, you can also think about the fact that we only have a litter of 3 puppies. And so it might have been due to chance that every single one of them came out with a big B, small b, and so they appeared black. But typically, you can say that, yes, the breeder might have been correct for this question. Now, I think this is the last thing for this video, and then we go on into more genetics, and that is multiple alleles. So in some cases, we have more than 2 alleles. So far, we've been dealing with 2 alleles for the same gene. We've said, oh, you're either black or brown. It's either white or black. It's either pink or yellow. In some cases, like things like blood groups, and I hope you all know your blood groups as you're watching this, but when we come to things like blood groups, we have multiple alleles. And that's because these alleles all influence what exactly happens with your blood group. So for example, you already know, or I hope that you know, that your blood group can either be A, you can either be B, you can be AB, or you can be O. Of all of these blood groups, O is the recessive one, rather. A and B are dominant, O is recessive. A and B can be co-dominant, and that is why you would find people who have AB. And O is always recessive, which means for a person to be an O blood group, they have only O alleles in their blood. Okay, and that's why you can see over here, this is also just to let you know how blood groups influence blood transfusions. So for example, if you have an A phenotype as your blood group, this is typically what your genotype looks like. You either are homozygous, so you have AA, or you are heterozygous, which means you have A and you also have O. The O is usually not shown, but O is recessive, so you can be AA or AO. And the same thing goes for B. If you're co-dominant, then it's just A and B that are coexisting, and if you're O, then it's just OO, because that is the only recessive allele for blood group. And obviously the antigens determine what happens when you're receiving blood. And if you haven't watched the videos on immunity, I suggest that you do so. But if a person is A blood group, then they have an antigen for A, and that means that when they want to take blood, they can only take blood from A and from O. Okay, because O doesn't have any antigens on it at all. So it means that when you give a person blood from an O from an O donor, then that blood is safe for them. But if you give a person blood from maybe B, for example, that can create problems and you would have like immune attacks and so on. Same thing goes for B, and then for AB is able to receive from A, from B. It can also receive from AB and it can receive from O, whereas O can give to everyone, but it can only receive from other O donors. I know that sounds a little bit unfair, but hey, this is life, right? So let's look at how that might play out in a question, because I'm sure for you, you're like, what is going on and why do they have subscripts or superscripts? Sometimes we write the alleles that way, and I'll try to do that with this particular question. So here it says a man of blood group B and a woman of blood group A have 3 children. One is group A, one is group B, and one is group O. What are the genotypes of these 5 people? When students have to do this question, the first thing I tell them to do is to look at what the children have. So you have a child that has A, you have one that has B, and you have one that has O, which suggests that at some point, because O is recessive, so for this child to be O, there are 2 O's in the parents. And so you can from there just easily guess that the man's blood group is IB, if I want to write it, IB, because he's blood group B, IO. Remember that O is recessive to B, so that means he would present as IB, okay? His wife in that case would then be, I think I can actually just do that here and make it a table. She would be IA and IO, okay? Because they have a child with blood group O, so there has to be 2 O's because O is recessive. And if we then look here at what the possibilities are, this is not the prettiest drawing, but what we would end up with here is IA and IB. Okay, and then here we would have IA and IO. And again, remember that we're crossing this times this, and then we cross this times that, all right? And that's how we're getting our results, in case you were a little bit confused. And then here we have IB and IO, and over here we have IO and IO. Okay, so in this case now, we can see that there's a possibility of making an offspring with an O genotype. I know that looks like a robot's eyes, but trust me, it's not what it is. It's OO over there. So we can see the possibility here, which means that for the man, this is his genotype, IBIO. For the woman, it's IAIO. And for their offspring, the one that's A, group A, is actually IAIO. And the one that's group B is IBIO, because those are the only possible combinations that you would get from this. All right, I hope that was clear. If it wasn't, again, just post something in the comments and I will respond as soon as I can. Or your colleagues might respond to you if they see your question before I do. Now let's look at the last question for the slide. And I know this video might have been a bit long, but I really hope that you're finding it helpful, that we are walking through the examples and it's helping you sort of get a hang of how genetics works. And so you feel more confident when you're giving questions to address it. So let's look at this question here. This is one of my favorite questions. It says coat color in rabbits is determined by a gene with 4 alleles. So here we're dealing with multiple alleles, again, 4 different ones. The allele for agouti is the normal coat, and it is dominant to all the 3 alleles. So let's try to do this. I'm going to use a black pen because I think the red one is just a little bit too much sometimes. So A is equal to agouti. I'm just going to write that here. And then it's dominant to all the other 3 alleles. The allele for albino coat is recessive to all the other alleles. So we can actually just say small a is albino. I'm just going to write it as ALB. Then we have the allele for chinchilla, which is dominant to the allele for Himalayan. So it's agouti over all of them. And then agouti is dominant to chinchilla, which would be C. And chinchilla is dominant to Himalayan, which would then be H. Okay, so we can just sort of use that. And it says here, write down the 10 possible genotypes for coat color and their phenotypes. Of course, this is a nightmare for many students because they say, excuse me, how do I even start to do that? But this is not difficult at all. So we know that agouti is dominant to all the other ones. So we know that in that case, we can have capital A, capital A, which is homozygous dominant for agouti. We can have capital A and C, which is still an agouti rabbit because agouti is dominant to chinchilla. We can have A and H, which is still agouti because it's dominant to Himalayan. And we can have A and small a. Okay, these are all the possible agouti genotypes that we can have. Then now let's look at the chinchilla genotypes. We can have big C, we can have CC, never mind big C. We can have big C, we can have CC again, which just tells us that this is a chinchilla rabbit. We can also have CH because chinchilla is dominant to Himalayan, and we can have CA because chinchilla is dominant to albino. Okay. I hope this makes sense. So basically, I'm taking all the different possible coat colors and writing the genotypes that are possible for them based on these 4 different alleles. Then let's look at Himalayan. So we can have Himalayan in terms of it looking like that, or we can have Himalayan that is dominant to albino. Okay, and then we can also have albino. So let's see, how many do we have at the moment? We have 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. And that is the answer to that question. Very easy. Again, if you have a question like this, what are the possibilities? Which one is dominant to all of them? So agouti dominant to everything else. And so you can just write out agouti as the dominant for all of them. Chinchilla is dominant to Himalayan, but always remember that they also have to have their own homozygous traits. Okay, then I'm just going to do question 1 here because I don't want this video to be excessively long. And you can try to do questions 2 and 3. So if you can just pause the video after I'm done and sort of look at those 2 questions on your own and post the answers in the comments so that your colleagues can see what you've been up to and they can correct you if you're wrong. All right, so here it says draw genetic diagrams to explain each of the following. An albino rabbit is crossed with a chinchilla rabbit, producing offspring that are all chinchilla. Okay, and then 2 of these chinchilla offspring are then crossed, and they produce 4 chinchilla offspring and 2 albinos. And so what obviously would that look like? So let's think about this again. It says an albino rabbit. We know that the albino rabbit or the albino coat color is recessive to all the other alleles. So the only possible albino genotype that we also have here is small a, small a. So we can just write that over there and just say small a, small a. All right, and it was crossed with a chinchilla rabbit. So chinchilla, we can just write as C and C. All right, and of course we are assuming here, right? So we then have CA, 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 CA. Now, because chinchilla is dominant to albino, all of these will be chinchilla. Then it says 2 of these offspring are crossed and they produce 4 chinchilla offspring and 2 albino. So basically you then have CA being crossed with CA. And in this case, you end up with CC, which is fully chinchilla. You end up with CA, which is still chinchilla, but heterozygous. You end up with CA over here, again chinchilla, and you end up with AA, which is what you would call your albino. And that, I believe, answers that question. So yes, just for homework and for practice, just do 2 and 3 and see what you come up with. And if you feel confused in any way, then just post a question in the comments, and I'm sure your colleagues will get back to you, or if I see your question first, then I will get back to you. Until the next video where we continue with more crosses, have a good time.